Hi learners, welcome back. I am Dr. Gurunathan from Kristu Jayanti College and I welcome you today for the topic on genome sequencing. So what is sequencing, or in general what is DNA sequencing? It is the process of determining the sequence of nucleotide bases in a piece of DNA that is given to us. When you speak about determining the sequence of nucleotide bases, it speaks about the presence of adenines, thymines, cytosine and guanines. And you can see that how are they arranged, how these are orderly arranged. Understanding this is so vital, and that process is called as sequencing, or sometimes commonly referred as DNA sequencing. And in today's scenario, with the right equipment and the materials, sequencing a short piece of DNA is relatively straightforward. Whereas when you're going to sequence the entire genome of an organism, what I mean here about an entire genome is the entire content of an organism's DNA. That remains a complex task, and that requires several steps, which begins with breaking of the DNA in the genome into smaller pieces, then sequencing those pieces and assembling them back into a single long consensus sequences. So you see that this method has been commonly employed in many different projects, and particularly it has been employed in human genome project, because of which the cost has also come down, and also there has been a very big advantage when you speak about applying these techniques to the human genome project, where the results were obtained much quicker and much faster. So what is genome sequencing? As I said earlier, in DNA sequencing, the focus is understanding the order of A's, G's, C's and T's arranged in a given DNA fragment. If I am doing the same thing for an entire genome of an organism, we call it as genomic sequencing. And we are all aware about the presence of genome of an organism, which is made up of unique DNA or RNA sequence. And whole genome sequencing, which is in short referred as WGS, determines the order of all or most of the nucleotides in the genome. So the information which is encoded in the genomes of disease causing microbes such as bacteria, viruses and fungi will provide unique genetic fingerprints to the researchers, so that that can be further studied, further analyzed and further exploited for various aspects. And when you speak about genome sequencing, we are also focusing on an area called next generation sequencing. And this speaks about the sequencing technologies that are currently available, which can process a large quantity of genetic material at any given point of time. Even though these technologies are available since 2004, they are very popular in the recent times, because they have not only replaced the previous method, which is called as the Sanger sequencing, but they are also having the advantage of high throughput, particularly high throughput whole genome sequencing. And you see that newer sequencing platforms are currently available, which has revolutionized this field by generating larger volumes of data and dramatically lowering the cost of sequencing. Moving down to the steps that are present in genome sequencing, there are many steps available. The step 1 begins with extracting the genome which is to be studied. And this genome is basically strands of DNA or RNA which are present in the microbes or in higher organisms. Particularly when you speak about the RNA molecule, it is very commonly seen as a genetic material in viruses, and in case of studying the transcriptome of the organism, then we would like to go for studying the mRNA molecule. Step 2 involves library preparation, which focuses on preparing the DNA that has been extracted. Particularly, we need to break the DNA into smaller pieces, or we might require the RNA to be converted into a double-stranded DNA, or sometimes you have only single-stranded DNA in the source cells. So in that case, we need to convert that to double-stranded DNA, because a double-stranded DNA is required for sequencing to happen. Apart from these two aspects, we are also adding to the ends of the fragments that are produced by materials which are called as adapters. And when you add these adapters to the ends of the fragments of the DNA which is obtained from the source cells, we are preparing what is called as the library, and now it is ready for sequencing. So that takes you to the next step, which is called as sequencing, and the library is loaded into a sequencer, which is a machine which can identify the nucleotide bases which are present in the DNA fragments. And several next generation sequencing devices, they work upon the fluorescent signals that are being emitted during the process of reading each of these bases. And there are other sequencing platforms which monitor changes based on the electric current which is generated as the DNA passes through tiny holes. And this step will take a day or two for completion. And the last step is analysis. So this stage is where you require the support of computers more importantly than the other three stages. In fact, the other three stages, they depend more on the wet lab output, and they depend upon what kind of DNA molecule or the RNA molecule that you're going to use for the sequencing. Whereas the fourth step, it requires a lot of effort, and a computational support is definitely required in this step. And as you can see here, the sequencer produces data which is millions of long strings of letters that are then assembled together or aligned with a reference sequence. And you see that when this happens, we are having with us the genome of the organism that we are intending to study. And this can take days, sometimes even weeks, or in few cases it can even go up to months. So this is just a pictorial representation of the whole genome sequencing using a bacterial genome. As you can see here, you have a bacterial culture from which the DNA is extracted and purified, and that DNA is broken down. We call that step as DNA shearing. Once the DNA is sheared, a DNA library preparation is initiated, where you're going to perform the addition of adapters. And once you have the adapter added DNA fragments ready, we are going in for what is called as sequencing. So the library that is being prepared is subjected to sequencing with the help of the sequencers which are available there. And once you have got the sequencing done, you get the data. So the sequencer produces millions of DNA readings which are then subjected to DNA sequence analysis to find the final outcome. Moving down to the next step, that is shotgun whole genome sequencing. This is a special approach in whole genome sequencing, and this approach has been quite useful in several genome projects that are underway. Particularly, this approach has been very popularly used in human genome project, where the first step begins with multiple copies of the genome being purified, and then they are subjected to random fragmentation by ultrasonication. And this will lead you with a mixture of overlapping fragments of varying sizes, which are then segregated using agarose gel electrophoresis, and based on the sizes we are selecting the fragments. Once the fragments are selected, as you can see here in the picture, there is a 10 kb fragment and there is also a 2 kb fragment. So according to the size of the fragment, we are making a library. We are building up a library, and once the libraries are built for each of the sizes that is existing here, they are then going for mate pair sequencing. And once the mate pair sequencing is done by random selection of each of the libraries, we are analyzing them and we are preparing what is called as contig preparation. And once contig preparation is done, then we go for what is called as what is called as then we go for what is called as the assembly of the sequence, and finally we end up with our output. And there is also another approach which is called as clone by clone sequencing approach, which is also employed, and this approach is less popular compared to your shotgun whole genome sequencing. And this involves usage of several artificial chromosomes. Particularly the bacterial artificial chromosomes are more commonly employed in clone by clone sequencing approach. With this, I end my session today. Thanks for your attention. Take care. Bye bye. We'll meet in another interesting video.